Hi guys, I hope you all are doing great. Welcome you all to my YouTube channel BIOSKUL. BIOSKUL, it will focus on subjects of life sciences. This channel will be helpful for BSc, MSc and those students who are preparing for life science competitive exams. In today's session, we will focus on fertilization in sea urchin. Fertilization in sea urchin, it is an important topic in view of CSIR-UGC NET exam. As it is a large chapter, we will discuss it in three parts. The part 1, part 2 and part 3. The part 1 it will focus on chemoattraction and the acrosomal reaction in sea urchin. At the end of the slide, we will discuss about the previous year CSIR-UGC NET questions. So let's get started. What is fertilization? Fertilization is a process whereby the two sex cells, the gametes like the sperm and the egg, they fuse together to create the new individual which has the genetic potential that derived from both the parents. As here you can see that the haploid sperm fuse with the haploid egg that leads to the formation of the diploid zygote. Yes, in this chapter we are focusing on the fertilization in sea urchin. So let's know what the sea urchin is. The sea urchin, it is a spiny globular animal, belongs to the phylum Echinodermata, as it looks like the porcupine, so it is called as the porcupine of the sea. Sperm of sea urchin. This is the structure of the sperm. The sperm has basically the three important parts. The first one is the head, the second one is the middle piece, and the third part is known as the tail. In the head of the sperm has the two important structures. The first one is the nucleus. A haploid nucleus is present, and the anterior portion of the haploid nucleus there is a cap-like structure, as here you can see. This cap-like structure is called the acrosomal vesicle, and this acrosomal vesicle it is filled with the proteolytic enzymes, and the proteolytic enzymes and the exocytosis of the proteolytic enzyme that help in the process of fertilization. We will discuss more about it in our coming slides. And the second part of the sperm, it is the middle piece. In the middle piece, it possesses the numerous mitochondria. These mitochondria produce the energy for the movement of the tail. And as you know that the sperm it is a motile cell, and how the sperm is the movement of the sperm it requires the energy, and the energy is provided by the mitochondria. The mitochondria it is present in the middle piece. The next is the structure of the egg. This is the structure of sea urchin egg. In the structure of the sea urchin egg, here you say in the gray color is the plasma membrane. We know that plasma membrane it is a semi-permeable membrane, and the semi-permeable membrane it has a property that it will allow the selective ion and the substances to enter into the cell or from the exit from the cell. And outside the plasma membrane, there is a vitelline envelope. The vitelline envelope, it plays the important role in the sperm-egg binding. And outside of the vitelline envelope, there is a jelly layer. The jelly layer, it helps in the species-specific sperm-egg recognition. Okay, so the vitelline envelope, it helps in the sperm-egg binding, and the jelly layer, it helps in the sperm and egg recognition. We'll discuss about it. Then in the inside of the plasma membrane there is a cortex. It fills with the cortical granules. As you see the red dots are the cortical. These red dots are the cortical granules. The cortical granules are derived from the Golgi complex, and this structure is homologous to the acrosomal vesicle of the sperm. The important point is that the acrosomal vesicle of the sperm and the cortical granules of the egg both are derived from the Golgi bodies. The sperm the sperm contain only one the acrosomal vesicles, whereas in sea urchin the egg contain approximately about 1500 cortical granules. Okay, so how does sea urchin egg look like? The sea urchin egg it has a plasma membrane. The plasma membrane is covered by the vitelline envelope. The vitelline envelope it helps in the sperm egg binding, which is covered outside by the jelly layer, which is helping the species specific sperm egg recognition. Inside the plasma membrane, there are the cortical granules. The cortical granules are homologous to the acrosomal vesicle of the sperm. The acrosomal vesicle of sperm and the cortical granule both are derived from Golgi bodies, and they contain the proteolytic enzyme. Now, what is the function of the cortical granule? The cortical granule, the main function is to prevent the polyspermy. Its main function is to prevent the polyspermy. We will discuss when we go to the our polyspermy, prevention of polyspermy slide. Next comes to steps of the fertilization. What are the steps of fertilization? The fertilization has 4 steps. The first one is the recognition of egg and the sperm. The second is the fusion of sperm and egg membrane. Third one is the prevention of polyspermy, and the fourth one is the metabolism of egg. So, first we will focus on the recognition of egg and sperm. This is very important. This is very important chapter. Uh, and you will definitely get a question in CSIR net exam or in many life science competitive exams. The recognition of egg and the sperm, it basically has the 5 steps. You have to remember the steps in sequence wise. So the first step, what's happened in the first step? In the first step is the chemo-attraction of sperm to the egg. The first step is the chemo-attraction of sperm to the egg by the soluble molecule which are secreted by the egg. So the first step is the chemo-attraction of sperm toward the egg, and how they are chemo-attracted? The chemo-attracted by is chemical molecules secreted by the egg. Then the second step is exocytosis of acrosomal vesicle. The second step is exocytosis of acrosomal vesicle. Third step is the binding of sperm to the extracellular envelope of egg. The fourth step is passing of sperm through this extracellular envelope, and the fifth one is the fusion of egg and the fifth step is the fusion of the egg and the sperm cell plasma membrane. The chemoattraction of sperms toward the egg, which is the first step of sperm and egg recognition. How the sperm are attracted towards the egg? As we know, the fertilization is of two types. One is the external fertilization and another one is the internal fertilization. The sea urchin, it lives in the sea water, so it has the fertilization in sea urchin is external. The fertilization in sea urchin is external type of fertilization. In sea urchin, the sperms are attracted towards the egg by the process is chemotaxis. Okay. In the chemotaxis process, a chemical molecule is secreted by the egg. Then what is that chemical substances? The chemical substances is called as the result. The chemical substance it is called the result. It is very important. The chemical substance is called the result. Result it is a 14 amino acid peptide. It is a 14 amino acid peptide. Where it is isolated? It is isolated from the egg jelly of sea urchin. So, what is the chemoattractant molecule? The chemoattractant molecule is the result, and the result it is a 14 amino acid peptide. It is isolated from the egg jelly of the sea urchin. A very low concentration of result in the seawater can attract the sperm of its own species. It will attract only the sperm of its own species. That means the chemo attraction is species specific. Okay. This act is act as a ligand, and it binds to the receptor present in the plasma membrane of sperm. Okay, the result it will bind to the plasma membrane of the sperm. It is a sperm activating peptide which causes the increase in mitochondrial respiration and the sperm motility. It causes the increase in mitochondrial respiration, also the sperm motility. So what are the points we should remember? The point we should note is the fertilization in sea urchin is external. The sperm attracted towards the egg by a chemical molecule secreted by the egg jelly, which is called the result. Result it is a 14 amino acid peptide. The result it will bind to the receptor present in the sperm plasma membrane. When the result bind to the sperm plasma membrane, it increase the mitochondrial respiration and the sperm motility so that the sperm is attracted or moves towards the. So here in the diagram you see the result. Result, it is secreted by what? It's secreted by the egg jelly coat, and it binds to the receptor present in the sperm plasma membrane. Okay, the ligand bind to the receptor. When the result bind to the receptor present in the sperm plasma membrane, it activate the, what will be get activated? The receptor. In the receptor there is a RGC. RGC is the receptors guanylyl cyclase. The receptors guanylyl cyclase, then it will activate the or convert the GTP to cyclic GMP. Okay, it is a cell signaling process like a ligand bind to the receptor. When the ligand bind to the receptor, it activates the guanylyl cyclase. The guanylyl cyclase enzyme, it convert the GTP to cyclic GMP. When the cyclic GMP get activated, it open the calcium channel in the plasma membrane. It open the calcium channel in the plasma membrane that leads to the influx of calcium into the sperm, and that help in the swimming of sperm towards the egg. Okay, so what happens first? Result, it is secreted by the egg jelly coat of sea urchin that bind to the sperm plasma membrane, bind to the receptor of sperm plasma membrane. In the sperm plasma membrane, it activates the receptors guanylyl cyclase. Then the guanylyl cyclase, it converts the GTP to cyclic GMP. Cyclic GMP, it opens the calcium channel in the sperm cell membrane that allow the influx of calcium, and the influx of calcium activate the sperm motility, and the sperm swim off the research gradient towards the egg. Then the next step is the acrosomal reaction. So the acrosomal reaction, it has the two parts. The first part is the fusion of acrosomal vesicles with the sperm plasma membrane. The second step is the extension of acrosomal process. So we will discuss about the first step. First step is the fusion of acrosomal vesicle with the sperm plasma membrane. As in the structure of the sperm, you see in the head of the sperm, the head part of the sperm, it has a haploid nucleus, and above that haploid nucleus there is a cap-like structure that is called the acrosomal vesicle, and this acrosomal vesicle, it is filled with the proteolytic enzyme, and it is derived from the Golgi body. So when the acrosomal vesicle, it fuses with the sperm plasma membrane, the acrosomal reaction in sea urchin, it is initiated by the contact of sperm with the egg jelly. When the sperm get contact with the egg jelly, as we discussed by the process of the chemoattraction, that allow the calcium ion to enter into the sperm head. The calcium ion will be enter into the sperm head. Then this calcium mediated the fusion of acrosomal membrane with the sperm plasma membrane. When the calcium ion get influx into the sperm plasma membrane, that lead to the fusion of acrosomal membrane with the adjacent sperm plasma membrane, that leads to the exocytosis of acrosomal vesicle. When the acrosomal vesicle exocytized, then that release the proteolytic enzymes. This enzyme digests the jelly coat till it reaches the egg surface. So in the first step, what the first step means? The first step is the fusion of acrosomal vesicle with the sperm plasma membrane. When the acrosomal vesicle fuses with the sperm plasma membrane, it is initiated. There is a contact between the sperm and the egg jelly membrane. When there is a contact between the sperm and the egg jelly, that lead to the influx of calcium ion. The calcium ion mediate the fusion of acrosomal vesicles with the adjacent sperm plasma membrane, that lead to the exocytosis of acrosomal vesicle and release of proteolytic enzyme. That proteolytic enzyme digests the jelly coat and the sperm reaches the egg surface. And the next step, what is the second part of acrosomal reaction? The second part of acrosomal reaction is the extension of acrosomal process. Okay, so how the acrosomal process, the acrosome is get extended. It is extended if that involves the polymerization process. In polymerization process, what happens? The globular actin, which is present in the acrosomal region, it converted into the filamentous actin. Okay, so the extension of acrosomal process, it is a polymerization process where the globular actin, it get polymerized into the filamentous actin. Okay, the filamentous actin is the form the basis of acrosomal process, which protrudes from the head of the sperm, and that protrudes from the head of the sperm. The tip of the acrosomal process is covered by a protein. That protein is called as the bindin. Okay, the tip of the sperm, the tip of the acrosomal process, the tip of the acrosomal process or the acrosome of the sperm, it is covered with a protein. That protein is called as the bindin. It is important. You have to remember it. What is the protein name? The protein name is bindin. Where it is present? It is present in the acrosome of the sperm. And what is the function of the bindin? Is the function is help the sperm to bind with the egg surface. Its function is to bind the sperm to the egg surface. So the bindin, it is act as a ligand, so it should be bind to a receptor on the egg surface. The vitelline envelope of plasma membrane of egg have a species specific binding receptor that is called the EBR1. This bindin, which is present in the acrosome of sperm, it binds to the receptor present in the egg plasma membrane. That receptor is called the EBR1. What? The sperm acrosome has bindin. The sperm acrosome has the protein. That name is called the bindin. Bindin binds with what? Bindin binds to the EBR1. Bindin binds to the EBR1. Where the bindin is present? The bindin, it is present in the sperm. It is present in the sperm. Whereas it is present in the sperm, and the EBR1 we will find it in the vitelline membrane or the vitelline membrane of egg. It will find in the vitelline membrane of the egg. It is important. Okay, so this is the diagram. In this diagram, as you can see, this is the structure of the sperm head. Here the blue part is the nucleus, and there is a globular actin and the acrosomal membrane. So the first part of the acrosomal reaction is the fusion of acrosomal membrane. Fusion of the acrosomal membrane with the sperm plasma membrane. So when the acrosomal membrane and the sperm plasma membrane get fused, when the sperm and egg they come in contact with each other, and that lead to the influx of calcium ion. That calcium ion mediate the fusion of acrosomal membrane and the sperm plasma membrane. In the next step, what happens when the acrosomal membrane and the sperm plasma membrane they get fused? That leads to the exocytosis of the acrosome. That lead to the exocytosis of acrosome. And the exocytosis of acrosome that releases the different type of the proteolytic enzyme. Here you can see the proteolytic enzymes are get released. When the proteolytic enzymes are get released, that help in binding of the acrosome, that help in lysis of the egg jelly membrane and jelly core, so that the sperm it come in contact with the egg plasma membrane. And the second step of acrosomal reaction is the extension of acrosomal process. Here you see there is a globular actin present, and the extension of extension of acrosomal process, it involves the polymerization of globular actin to the filamentous actin. Here the globular actin is start to the polymerize to form the filamentous actin, and when the filamentous actin is forms, then the acrosomal process it protrudes out from the head of the sperm. This is the head of the structure. Now is the acrosomal process, it protrudes out from the head of the sperm. And that acrosomal process or the acrosomal process of the sperm, it has a protein. The protein is called the bindin. It has a protein, it is called the bindin, and this bindin protein, it binds to the its receptor is called the EBR1. It binds to the receptor. The receptor is called the EBR1. The receptor is called the EBR1, and EBR1, it is present in the vitelline envelope or the plasma membrane of the egg. It present in the vitelline envelope and plasma membrane of the egg. Hope you get a clear. So the next side will be the binding of sperm to the egg. Okay. So when the lysis of vitelline envelope is followed by the fusion of sperm plasma membrane with the plasma membrane of the egg. Okay. So the binding of sperm with the egg plasma membrane, it appears to form the fertilization cone in the egg. Like when the exocytosis of acrosomal vesicle takes place, that leads to the lysis of the vitelline envelope of the egg because of that proteolytic enzyme. So that lead to the binding of the sperm to the egg, and this fusion, it will form the fertilization cone in egg. The fertilization in cone in egg is like that of the acrosomal process, and it is formed by the polymerization of actin. When the sperm it binds to the egg, then egg it form a fertilization cone. The fertilization cone, it is formed by the polymerization of the actin molecule. The one important question is that how the sperm get fused with the egg. Both are the antigenically different. The sperm plasma membrane and the egg plasma membrane, they are antigenically different from each other. But however, it becomes the incorporated into the plasma membrane of the egg as a mosaic patch. They are incorporated, the sperm plasma membrane incorporated into the plasma membrane of the egg as a mosaic patch. Okay. Then the next question is how the sperm plasma membrane get fused with that of the egg plasma membrane. Yes, that is mediated by a specific protein. That specific protein is called the fusogenic protein. Then what the fusogenic protein? The fusogenic protein, the function is to fuse the plasma membrane of the two cells. Okay, the fusogenic protein here, it help in the fusion of the sperm plasma membrane with the egg plasma membrane. One of the fusogenic protein is bindin. Bindin is the fusogenic protein, and where it is present? It is present in the acrosomal process of sperm. Okay. Then, so here you complete the acrosomal chemoattraction and the acrosomal process, as well as the binding of the sperm with the egg. Hope you have a clear idea about all of these chapters, all of this topic, like how the sperm chemo-attracted towards the egg, then how the acrosomal reaction takes place, and how the sperm and egg membrane, they fuse together. In the next slide, or in the next video, we'll focus on the prevention of polyspermy in sea urchin. Okay. Now we'll discuss on some previous year CSIR-UGC net questions. Okay. So here is the first question. What is the question? The question is membrane bound Golgi derived containing proteolytic enzymes in sperm of sea urchin are called. Okay. Hope you all know the answer, and obviously the answer will be the acrosomal vesicle. As you know, the acrosomal vesicle, it is rich in the proteolytic enzyme, and it is also derived from the Golgi complex, and it is present in the sperm of sea urchin. Okay. Then the next question. Successful fertilization in sea urchin. The successful fertilization in sea urchin, it demands the specific interaction between proteins and receptors of sperms and egg. Okay. We have already discussed the fertilization. It requires the specific interaction between the proteins and receptors of sperms and the egg. In view of the above, which of the following combination is correct? Okay, so what is the first option? The option A, bindin in acrosome and bindin receptors on egg vitelline membrane. Okay, the bindin, where it is present? Obviously, it is present in the acrosome of sperm, and the bindin receptor on egg vitelline membrane. What is the bindin receptor? The bindin receptor is the EBR1. So it seems to be the correct answer. Okay, so for the successful fertilization, it leads the interaction between the sperm and egg, and how the sperm and egg are interact, or how the sperm and egg bind, become the binding which is present in the acrosome of the sperm. It binds to the receptor, that is the EBR1, which is present in the egg's mytilene membrane. So it will be the correct answer. Okay, let's see the second option, B. Binding in egg membrane. Binding, it is not present in the egg membrane, it is present in the acrosome of sperm, so it will be the wrong. Research on egg jelly and binding. The research is function is to recognize the specific sperm and egg. It has no role in the binding or in the interaction, so that's the wrong option. The proteasome on egg membrane and complex sugar on sperm membrane. So this one is also not the correct option. So the correct answer is binding in acrosome and binding receptor on egg vitelline membrane that demand the specific interaction between protein and receptors of sperm and egg. Okay. Then the third question. For successful fertilization in sea urchin, in interaction between the surface of the egg and acrosomal protein, specially a 30.5 kilodalton protein called bindin is necessary. Yes, it is necessary. The following factors could affect this interaction and prevent fertilization. Okay, so for the successful fertilization, interaction between the surface of egg and acrosomal protein, are both the interaction between the surface of the egg and acrosomal protein is necessary. So let's consider the first option. Removal of egg jelly polysaccharide. If you will remove the egg jelly polysaccharide, okay, then there will be no chemo attraction. Okay, so if there will be no chemo attraction, then how can we, a sperm will be interact toward the egg? So I think removal of egg jelly polysaccharide, it will affect the interaction of sperm and egg. So it's essential. Removal of binding receptor on egg vitelline membrane. Egg vitelline membrane. The egg vitelline membrane. Okay, for the fertilization, as we discussed, the bindin present in the acrosome of the sperm, it should bind with the bindin receptor on egg vitelline membrane. If we remove the bindin receptor on egg vitelline membrane, then it will also affect the interaction. It will prevent the fertilization. So the second option is also the correct. Okay, so here you got our answer. The answer is both the P and Q are correct. So both the P and Q are correct. So the option C is the right answer. Then the fourth question. Fourth question, in case of the sea urchin, which of the following is the correct sequence of event taking place during the interaction of sperm and egg? Okay. During the interaction of the sperm and egg, what are the steps takes place? Okay, in previous lines we have discussed there are the 5 steps. Now, what is the first step? The first step is the chemo attraction. In sea urchin, the first step will be the chemo attraction. Then second step is the acrosomal reaction. Third step is the binding of sperm to the egg membrane. Fifth step is the fourth step is the passage of sperm through the extracellular matrix. And fifth one is the fusion of egg and sperm cell membrane. So here the correct answer is option A. Option A is the chemo attraction, the first one. Second point is the exocytosis of the sperm and the egg membrane. Then the third step is the binding of sperm to the extracellular membrane of the egg, and the fourth one is the passage of sperm through the extracellular matrix, and fifth one is the fusion of fifth step is the fusion of the egg and sperm membrane. So it is the correct answer. So see the option B, it has a chemo attraction. Then the second step is the binding of sperm to the egg extracellular matrix of the egg. No, that's the wrong answer. And the third step is also same. The option C has also the first one is the chemo attraction, and the second step is the binding of the sperm to the extracellular matrix of the egg. That means this one is also wrong. Then the D. Option D is the chemoattraction of sperm to the egg by soluble molecules secreted by the egg. Yeah, that's right. Passage of sperm through the extracellular matrix. No, it's wrong. So the correct answer is the option A. Sorry, this one. This should be the D. By mistake, it will be it's the D. D. So thank you for watching. Let's learn together. If you find this video is more informative, if then please share with your friends who are preparing for CSIR NET exams, and please do like and subscribe my channel for getting more such videos.